So our today's topic for focus of the day is the genome sequencing. We'll see why is the news, what is genome, what is whole genome sequencing, advantages of genome sequencing, difference between gene and genome. Then we will come to our questions of prelims and the mains as well. Before starting, I want you to know that our coaching Analyst IAS is launching a super fast prelims batch. The batch will start from 15th of January itself, and the first 50 aspirants will avail 25% discount in their fees. This batch will cover your whole syllabus before the exam, and there would be regular test series to monitor your progress as well. So if you are interested, you can contact at our given number, or you can come directly at our office, which is at Old Rajinder Nagar of New Delhi. So we will see why certain news. So recently, researchers at the Indian Institute of Science Education and Research, Bhopal, have carried out whole genome sequencing of banyan and peepal from leaf tissue samples. What is a genome? So genome is the entire set of DNA instructions found in a cell. In humans, the genome consists of 23 pairs of chromosomes located in the cell's nucleus, as well as a small chromosome in the cell's mitochondria. A genome contains all the information needed for an individual to develop and function. The human genome contains about 3 billion nucleotides. What is a whole genome sequencing? All organisms have a unique genetic code or genome that is composed of nucleotides, bases, adenine, thymine, cytosine, guanine. Whole genome sequencing is a laboratory procedure that determines the order of bases in the genome of an organism at a single time. This includes sequencing all of an organism's chromosomal DNA as well as DNA contained in the mitochondria, and for plants in the chloroplast. What are the advantages of genome sequencing? So it provides a high resolution base by base view of genome. It captures both large and small variants that might be missed with targeted approaches. It identifies potential causative variants for further follow-up studies of gene expression and regulation mechanisms. It delivers large volumes of data in a short amount of time to support assembly of novel genomes. Whole genome sequencing also offers a better determination of copy number variations, rearrangements, and other structural variations. So here is the difference between a genome and a gene. So a gene is a segment of a portion of DNA molecule, whereas genome is the total DNA content in a cell. Gene code for protein. Genome itself cannot code for protein as it contains almost all the DNA. Gene consists of only few base pairs as it represents only a DNA segment, whereas genome consists of all the base pairs in a cell. The study of properties of genes is referred to as genetics. The study of properties of genome is referred to as genomics. An individual has thousands of millions of genes. An organism has one genome. Now we will come to our prelims question. So with reference to agriculture in India, how can the technique of genome sequencing, often seen in news, be used in immediate future? First, genome sequencing can be used to identify genetic makers for disease resistant and drought tolerance in various crop plants. Second, this technique helps in reducing the time required to develop new varieties of crop plants. Third, it can be used to decipher the host pathogen relationship in a crop. You can try to answer the following questions. You can pause the video as I have given the answer in the next slide in the rightmost corner. So now we will see our mains question. What are the research and developmental achievements in applied biotechnology? How will these achievements help to uplift the poorer sections of the society? This question came in the year 2021 itself.